I hope
it's good. Good. The sharing sounds good. Yeah.
Okay, so today I would like to talk about the bacteria and phage. The things I'm working on is more about the ecology than the evolution. But of course, if you deal with the bacteria, sometimes you cannot just avoid mutation happening while you're looking at them. So I will see a little bit of it. But so the main focus of what I'm interested is that basically how these bacteria can survive this very big attack of phage. Just the one slide. To mention that the phage is everywhere. The estimate says that there are 10 to the 30 bacteria cells on Earth, while the people say there are like roughly 10 times more phage particles on Earth, so it's a lot. And it takes care of the very large part of the biomass recycling on Earth, like the 90% of the biomass in ocean is thought to be microorganisms, while 20% of them are estimated to be killed by the bios every day. And also in the human guts, like most of the virus you detect is actually bacteriophages, so that also you can imagine that it's very important for the microbiome. So that I myself worked to some extent sort of how this phage can play a role in the bacterial community. So that for example, even if the bacteria themselves had the nutrient competition, so that there should be a competitive exclusion, if the faster growing bacteria has a phage that attacks them, then there will be a space for the slower growing bacteria to get in, so that you can actually, for example, construct a food web of the bacteria and the phage that contains many bacteria types of the many phage types, as long as you have roughly the same number of the bacteria types and phage types. But today I want to focus a little bit more of the thought of the physical interaction between the virus and the bacteria and the importance of them in the ecology. And to go into it, I first sort of want to recap a little bit how the virus works. So the simplest the phage that we know is so-called virulent phage or lytic phage. So that basically what it looks like can be this this capsid that carries the genetic material, and then the tail fibers that recognize the normally host-specific protein that is expressed on the surface, and then they will eject the DNA or the RNA, and then that will be copied into making many of their own copies, and then after a certain time, they will come out by making a whole of the host cells. And if it's for E. coli, then this time can take from 10 minutes to 1 hour or so. And then after that, maybe 100 virus can come out. But just one of them is enough to kill the host. So it's really the deadly viruses. And so in general, I'm curious how bacteria can survive such a deadly viruses. And it seems like some viruses noticed that it's not really sometimes a good idea to kill all the hosts that viciously. So some phages actually had evolved to be so-called temperate phage. You probably all know about the lambda phage. So what's the difference between the virulent phage and the temperate phage is upon infection, at a certain probability, these temperate phages, including lambda phage, can choose to express certain sort of repressor protein that will repress the lytic cycle to be expressed. And then once you get into this state, it's called lysogens. And then once you are in this lysogeny cycle, then the phage will actually replicate with the host DNA. And also that these repressor proteins provide the immunity against the same phage, of course, because this one is repressing the lytic pathway to be expressed. So if a new phage comes in, then they just don't do anything. So basically this is a phage that brings in immunity once in a while, and this has been interpreted as maybe it's the bed hedging of the phage. Basically, the phage had evolved so that it doesn't kill all the hosts if the number of the host is not that high that you need to keep some for the future. Of course, in addition, there are all other ways for the bacteria that they evolved to avoid the phage attack. What you actually most often see if you do the experiment is the resistant mutants. And normally when you say the resistant mutant in the bacteria and phage context, then that means the loss of phage receptor. So basically phage cannot get in anymore. And of course, those receptors are not there for phage, but for importing something else normally. So of course, this kind of the receptor mutants do cost in the fitness, so that there is some measurement of these the receptor mutants growth rate in different media, and typically they are slower than the wild type. But this is, of course, the one way to be resistant to the bacteria, to the phage. But of course, in addition, phages had evolved infinite number of the defense mechanisms, and we also keep finding new ones. They can make an extracellular matrix to trap phages. They have something called restriction modification system that can recognize foreign DNA sequence. They can have the abortive infection or TA system. Basically, what they do is upon infection, they die. And then, of course, there is this very famous CRISPR's immune system that they have a small chance to actually learn the virus sequence and then become truly immune to the phage attack. And so recently, so that of course these individual molecular mechanism has been studied a lot, but then the relatively recently there was some work that tried to see how these kind of the defense system plays in the sort of population dynamics context. And then there is a very nice work from 2018 by Payne et al. That sort of used this CRISPR system to see if that can actually provide a so-called herd immunity, so that if you mix the CRISPR expressing cells and sensitive cells and then put the phage in, then can they survive? The sensitive cells can also survive. And then they did see when the 99% are the immune, then the sensitives can be protected. So that was sort of very interesting work. Though that there was a bit of caveat for this particular system. They actually artificially constructed this herd immunity, the CRISPR system in this experimental system, so that for this particular setup, actually the infected cell of the immune cell actually stop growth so that they die. So it's a little bit more closer to the actually abortive infection setup. But so that when we saw this, we thought that's interesting. And then when we actually think again about this temperate phage that actually brings in true immunity, then maybe that also can give the herd immunity. So that we just did the simple experiment of basically the mixing the sensitive E. coli and the immune sort of from this lysogens immune cells, and then we made them attacked by a phage that will be repressed if there is this repressor proteins expressed. So that basically, if this phage infect the lysogens, then it will inject the DNA, but it cannot do anything, and then the bacteria will just happily grow. And then, but of course, if you put this DNA in the sensitive E. coli, then this will express the phage, and then it will kill. And this phage, we chose the mutant phage that cannot choose the lysogen by themselves, so that the number of immune cells does not grow through the infection. But then so that we basically mix them and then see if sensitive cells will survive. And then we did see that it does have the herd immunity. So that here what you're seeing is that this axis is initial sensitive cells fraction, and then this axis is the survival fraction at the end. And then you can see that they do provide the immunity, though maybe it's not very threshold, like it's more sort of the smooth curve that we have observed. And then just for the curiosity, we started to wonder what happens if you mix instead of this the immune cells, but what if we mix this resistant mutant, which happens very often, which it had actually lost the surface protein to even absorb the phage. So we did basically the same experiment using this resistant mutant. And then what we see is these crosses over here. So that basically no one survives if you mix the resistant mutant with the sensitive cells. So that it might be sort of, once you see the result, it might be obvious, but this is really telling us that the absorption of the viruses by immune cell is really, really important for the herd immunity to play for the bacterial population. So that basically these lysogens are sucking up the viruses, and that provide the very big protections for the whole community. And once we see this, the sort of this absorption of the phage is also happening all the time, and that this protection by the absorption of the phages should also happen among sensitive cells. So sort of when you actually take this photo of the bacteria that is attacked by phage, then you often see these huge number of the phages are actually infecting the cells. That this black and white picture, each white dot are phage. So this poor bacteria is really completely covered by the phage. So basically, phage cannot tell if the cell is already infected or not. So the one cell can actually absorb a lot of phages. And that can, for example, provide a very inhomogeneous attack in the spatial structured environment. So for example, if you basically simulate phage as I don't know if you could see phage as just diffusing sort of red dot that if it hits the target can be, if it hits the cell, then it will get absorbed. So that just let them diffuse and absorb in the simulation. And then, for example, if you construct some small cluster of the bacteria, and then let one of the cells burst, and then let the phages to diffuse and attack wherever they randomly hit, then normally actually their neighbor cells get hit by the hundreds of the viruses, and that basically gives the effective burst size of these infected cells to be so small, so that provide the protection for the population by sort of releasing the less viruses to the environment. And this will be a sort of even more sort of important when you have the very dense colony. So what we thought is, so that if you have this dense colony, and then phage needs to attack from the outside, then maybe it's very hard to get in, because there is these the infected cells that still takes, say, 10 minutes to 1 hour before they burst, and the virus cannot really tell if that cell is already infected, so they will reinfect them. So that if we actually try to infect the bacteria colony from outside, and then what you are seeing here is so the blue is a simulation of a growing cells, and then once it gets infected, it will become yellow, and then it becomes red, and then it will burst, and it will release the hundreds of phages. I will not show phage itself, but so what you see is that they need to eat the colony from the outside. And even funnier thing is that, so that if you do exactly the same simulation, but start with a little bit bigger colony, then this is what happens. So basically the cells inside are growing while the phage is trying to kill, but it will be limited on the surface because of this absorption by the infected cells. So basically it's a two-dimensional killing versus three-dimensional growth. So there should be a critical size above which the colony actually stay alive, even though the attack is happening on the surface. And also this also have a funny prediction that if you have a higher absorption rate, that will protect the colony better, because it's hard for the phages to infect to the core of the colony. So that if we try to simulate what is this critical sort of the survival size of the colony, and then if I change the absorption rate, then this critical size becomes smaller and smaller as the phage absorption becomes stronger. So somehow if you get attached phage by phage bigger, then it's actually the more protective for the bacteria. And we also try to test this experimentally, so that we grown the colony of the different sizes, and then basically sprayed a lot of phage. In this case, we used the phage called the P1 Vir, and this one is sort of relatively slow in the time between the infection and the lysis, so that should give sort of a lot of the protection for this multiple infection mechanism. And then we wait overnight to see if colony kept growing or not. And what we saw is that if we sort of had grown the colony for roughly maybe 7 hours and then put the phage, then it seemed like almost all of the colony had survived and kept growing after that to be a bigger colony. But of course, when we present this result, then the question we get is, isn't it just because you have more resistant mutant in the bigger colony? And that, of course, is true, that the bigger the colony is, I have a higher chance to have the resistant cells. So that we also went into these colonies, we picked the cells and then tested 10 isolates per colony to see if they are sensitive to the bacteria, to the phage or the resistant to it. And then so this is sort of all the statistics we got. But this one, so among 10 colonies we tested for each different surviving timing. These are the number of the colonies that the 10 cells we tested are all susceptible to the phage. So that means at least in these colonies, we believe the majority of the bacteria are sensitive to the phage. So it's not just the resistance that explain these statistics. But at the same time, we see like then there are also the similar number of the cells that sort of almost everyone's resistant, and we don't really know how we can understand these statistics. After this, we are actually trying to see the dynamics of this killing, also using the more virulent phage T4, which has a shorter lysis time, to see if they can still survive. So now what we are seeing is that when there is no phage, the colony will grow, and then we also put green fluorescence to it. And then when you put the phage on it, then you can see it's sort of the phase away, so that they are sort of, you can see they get attacked from the outside. And this is the attack on the bit bigger colony, and then maybe at least it is surviving a bit longer, probably. And but also the one interesting movies I wanted to show, this ones get attacked and then becoming dark, and then you start to see some green things coming up. We believe they are this resistant cells, and then now it gets selected, so they are making a new sort of the bumps coming out. But so we can also see in this context, these mutants are very localized, and in the spatial context. And we are trying to get some quantification of these killing dynamics by looking at this green fluorescent and the bright field data, and then the data seems to be consistent with this. They are trying to kill from the surface, and so that they are becoming darker from the outside. It seems like with T4 cells, in the end, all the colony seems to be becoming dark and all killed, so that this the protection mechanism seems to be require a lot bigger colony than that we have been trying to do. But so that's basically the end of what I prepared. But so the main message is that the absorption is very effective way to inactivate viruses, and that matters a lot for the bacterial system where there are a lot of cells. Also maybe if I imagine a little bit bigger, then maybe this could also mean something for the viral spreading, for example, in tissue. Basically, if you had the tissue that has a lot of receptor for that virus, then maybe actually the infection is more localized because they may not be able to diffuse far away. So that kind of thing could be something one can think about by looking at these kind of behavior. Yes, thank you very much for your attention.
Thanks a lot for this great talk. So I don't see question yet in the chat. I see Joachim has a question. You want to start?
Yes, I was just wondering, I mean, what are the consequences of this absorption for the selection of, for example, virus burst size? So you know, do you think the viruses could sort of counteract that by making the bursts even bigger, or what is, you know, how should they deal with this problem?
So it's also that's actually very good question. I want to know, and also how can I say? So for example, the one interesting thing that the coming up now is, for example, some phages, when especially these temperate phages, they may try to actually regulate the receptor expression level of their host. So maybe they could sort of do that kind of things, and but also so this absorption becoming a loss is, how can I say? Yeah, it's also in a sense the unavoidable consequence of what phage can do, and for the burst size, of course, the bigger the burst size, the better. But there is again another trade-off, because the rate of the phage production per cell is, of course, there is a much so that they need to, if they wait long, they can make bigger burst size, but that's also.
Yeah, but because the absorption means that a lot of the burst is simply wasted, right? Yes. Because it just goes into the next cell. So then you might want to maybe have smaller bursts, but quicker, for example.
Yeah. So that they could try that. Yeah. And in this case, that will be better scenario for them. Yeah.
Thank you.
I was wondering, I mean, in terms of evolution, it is also favored to have a very high specificity, because if it sticks to things that are, I mean, to off targets, if they're in an environment with a high density of over targets, it can be problematic. I don't know. I wonder what is your opinion about what your results imply for, I don't know, dynamics of phages and bacteria in the gut, for instance, where you have a high density of bacteria?
Yeah, I think definitely they are very specific as far as I know, and also some of the bacterial protection strategy is sometimes like express a decoy receptor that sort of is not really connected to the inside, but phage can waste their DNA and so on. So that definitely the phage want to be specific, and then bacteria want it to be not very specific, and so that there is also a lot of arms race around it.
Is there more questions? I mean, I think I have more questions, but I. Okay. Yeah, I mean, interested. And do you think that, for instance, for bacteria to then, I don't know, for instance, to stick in biofilms or things like this could be also an adaptation against phages? Or?
Yeah, definitely. There is a very interesting work by the Knut Dretcher in Germany that he had grown the sort of the very big biofilm, and then when this biofilm is big enough, then they also have this sticky material around that can actually stick to the phage, so the phage cannot get in anymore. So that it definitely does work as a protection against phage.
Oh, cool. That's very cool. Okay, so if there's no more question, let's thank everybody of the session. It was a great, thanks a lot. And if you want to join us on Gather, we can have more discussion, but thanks a lot. It was very cool. Bye.